We should now ask how DNA encodes the properties of an organism. This question haunted biologists for a long time. On the one hand, we have an organism's genotype, or its precise makeup of DNA. We know that its genotype ultimately determines its phenotype, based on experiments like Avery's and many others. The question is how? What precisely is it about DNA that determines phenotype? The answer came from a couple of microbial geneticists, Beadle and Tatum, who formulated the one gene, one enzyme hypothesis. It states that genes act by encoding enzymes. Even in their day, it was clear that this was an oversimplification, because not all proteins are enzymes, and some enzymes are made of several proteins. A better way to formulate the idea is to say that genes encode a polypeptide. Even this is an oversimplification, but it will serve us very well for now. That is what this picture is showing. DNA in the nucleus contains information, and that information influences the properties of the organism, ultimately by coding for protein. DNA acts through an intermediate molecule of RNA, which serves as a message from the nucleus to the ribosome with the instructions to make a protein. Proteins perform all of the organism's functions and therefore determine its properties. This should all look familiar to you. It's a restatement of the central dogma of molecular biology. Gene expression is the process of making protein based on DNA sequence, and it comprises the processes of transcription and translation. Transcription is making RNA based on a DNA template. The most important RNAs in protein expression are mRNA, tRNA, and rRNA. There are others, but they're not part of our discussion right now. mRNA is messenger RNA, and when you think of a gene that encodes a protein, it actually encodes mRNA, and mRNA in turn encodes a protein. Transfer RNA, or tRNA, has three jobs. It binds the amino acids that will be built into protein, it brings those amino acids to the ribosome, and it reads the genetic code of the mRNA, the codons, via an anticodon that is part of the tRNA. Ribosomal RNA, or rRNA, is the RNA component of the ribosome. As you know, the ribosome is the site of protein synthesis. It is a large complex machine made up of many RNAs and protein molecules. The RNAs that are a part of the ribosome are rRNA. They are a structural component of the ribosome, but they can also provide enzymatic activity. This is the only time we'll talk about an enzyme that's not a protein. RNAs with catalytic activity are called ribozymes, and it is a ribozyme that catalyzes the formation of peptide bonds in a newly synthesized protein. As we have defined it, a gene is a sequence of DNA that encodes a polypeptide. But how does DNA encode protein? The answer is the genetic code, and that's what's shown here. Before we go over it, let's look at what's shown here on the left. The first step is transcription, making RNA using a DNA template. The resulting RNA is complementary to one of the two strands of DNA. That's shown here, DNA at the top and the complementary RNA underneath. In this case, pink is complementary to yellow and blue is complementary to orange. The code in the sequence of RNA is read as triplets of nucleotides called a codon. Each codon signifies an amino acid to be incorporated into a newly synthesized protein. In other words, a codon is a triplet of nucleotides that encodes a single amino acid. The genetic code specifies which amino acid is encoded by each codon. Let's take a closer look at the genetic code. First, we'll learn how to read it, and then we'll look at some special features. First off, it is RNA that directly encodes protein, so the genetic code represents RNA sequence and uses U, not the T of DNA. The first letter of the codon is the nucleotide at the 5' end of the triplet. The second letter is in the middle, and the last letter is the 3' end of the triplet. Since 3 nucleotides make a codon, and there are 4 nucleotides, A, C, G, and U, there are 64 codons, 4 times 4 times 4. Look at the table and count them. It's 4 by 4 for a total of 16 boxes, and each box has 4 sequences in it for a total of 64 sequences. That's more than enough to encode the 20 amino acids. If a codon were only 2 nucleotides, there would only be 16 codons, not enough to encode the 20 amino acids. So a codon has to be 3 nucleotides. Let's look at the sequence ACU. Start on the left and find the letter A, third from the top. Now go right 2 columns and you're in the C column. The box has 4 sequences in it. The top one is ACU, and you can see that it encodes threonine, which is abbreviated THR. So the codon ACU encodes the amino acid threonine. Look closely and you'll see all 20 amino acids are represented by the code. In fact, all of them except for 2, methionine, met, and tryptophan, trip, are represented more than once. That means that the genetic code is redundant. More than one codon can encode the same amino acid. It's redundant but not ambiguous. A codon encodes one and only one amino acid. Let's look at some other features of the genetic code. The codon AUG, which encodes methionine, also signals the start of translation. It's the start codon. So as a ribosome reads an mRNA, it will look for that AUG and start making a protein there, beginning with the methionine. There are also 3 stop codons. When the ribosome sees a stop codon, it stops making protein. The final feature to know about the genetic code is that it's universal. That is, all organisms on Earth use the same genetic code. Not only do bacteria and humans have DNA, but they use it in the same way. A DNA sequence would encode the same protein in humans or bacteria. We take advantage of this fact and actually use bacteria to make human proteins for therapeutic use. Insulin, for example, is made by taking the human sequence for insulin and putting it into bacteria. The bacteria then make insulin that is purified and used to treat diabetics. There are some exceptions to the universality of the genetic code. Remember that mitochondria and chloroplasts have their own genomes, and those genomes encode a few proteins. Chloroplasts and mitochondria use something other than the universal genetic code. Transcription is the process of making RNA based on a DNA template, such that the resulting RNA is complementary to one of the DNA strands. This process is very similar to DNA replication. In replication, DNA polymerase reads the template sequence and incorporates the complementary nucleotide into the growing polynucleotide chain. Transcription is the same thing, but RNA polymerase is reading one strand of the DNA, the template strand, and making a new RNA molecule complementary to it. That's what the top diagram shows. The RNA being made, in pink, is complementary to the lower strand of the double-stranded DNA molecule. The RNA product then serves as a template for protein synthesis. Note that both the newly synthesized RNA and the top strand of DNA are both complementary to the lower strand of DNA. The RNA and the top strand of DNA are therefore identical to each other, except that RNA substitutes U for T. The lower illustration shows how this process is initiated. RNA polymerase, the whole big yellow blob, is the enzyme that reads the DNA template and synthesizes a new RNA molecule. +1 represents the first nucleotide to be transcribed, or the first nucleotide of the new RNA molecule. The sequences at -10 and -35 are upstream of the sequence to be transcribed, but are critical to getting RNA polymerase to bind to the DNA template and telling it where to start transcription. This example comes from bacteria. Similar things happen in eukaryotic cells. Accurate binding and positioning of RNA polymerase is critical. But in eukaryotes, the positioning of RNA polymerase is guided by a large group of other proteins called transcription factors. Overall, the process in eukaryotic cells is much more complicated, and we won't go into any more detail than that. Gene expression in eukaryotes is more complicated in other ways as well. Eukaryotic mRNA is processed before it can be used for translation. The mRNA in eukaryotes is made in several steps. The RNA that is first made is called the primary transcript, and it is very similar to the DNA from which it was made. Note that the primary transcript, like the DNA that encodes it, is made up of exons and introns. The exons are the active part of the gene that encode protein. The introns don't encode protein and would interfere with translation, so they have to be removed. The term intron comes from intervening sequence. That is, it's between the exons. The RNA is first made with the introns still in place, the primary RNA transcript. The introns are then spliced out. That is, they are removed and the exons are spliced together to form one contiguous protein coding sequence. Other processing of the RNA includes 5' capping and 3' polyadenylation. Both these steps improve the stability of the RNA. Without them, the mRNA would be rapidly broken down. The 5' cap and the 3' poly A tail also increase the efficiency of translation of the mRNA. RNA processing all happens in the nucleus where RNA is made. The fully processed or mature mRNA, or mature transcript, is then transported out of the nucleus into the cytoplasm where translation happens. The mRNA serves as a template for the production of protein. But how is the sequence of mRNA turned into a sequence of amino acids in a protein? The answer is tRNA, or transfer RNA. tRNA has three distinct jobs. First, it binds to a specific amino acid to form an amino acyl tRNA, AA-tRNA. Each tRNA has to bind the right amino acid because it is the job of the tRNA to make sure that the correct amino acid gets incorporated in the correct position of the growing polypeptide chain. The tRNA then travels to the ribosome with its bound amino acid in tow. Finally, at the ribosome, the tRNA binds to a codon of mRNA via the anti-codon loop on the tRNA, which is a 3-nucleotide sequence complementary to the codon of the mRNA. This once again highlights the importance of complementary base pairing. This system can only work if there's coordination between the anti-codon and the amino acid to which tRNA is bound. The enzyme that adds an amino acid to the tRNA has to read the anti-codon sequence and attach the correct amino acid to the tRNA. Now we come to the ribosome, where protein synthesis actually happens. Here is an overview of the process. On the next slide, we'll look at it in more detail. The ribosome itself is this big structure in the middle. Note that it is made of two subunits, a large in red and a small in white. Don't be fooled by this seemingly simple structure. Both subunits comprise many protein and RNA molecules. When the ribosome is not busy making a protein, the subunits exist separately, and they come together when translation begins with the mRNA binding to the small subunit. Here you see it already assembled, and the mRNA sits between the small and large subunit. Inside the large subunit, you can see a tRNA molecule, green, base paired with its anti-codon to a codon of the mRNA. At the other end of the tRNA, a growing polypeptide chain is attached, in blue, and you can see individual amino acids. The polypeptide will continue to grow one amino acid at a time until the chain is complete. That is, when the ribosome reads a stop codon in the mRNA. This festive picture shows you the ribosome in more detail and in the party hat mode. I guess this is where the Lady of Hats gets her name. It shows the ribosome in a cutaway view so you can see what's happening inside. Here you see two tRNA binding sites, the A site and the P site. There's actually 3 sites, but the third one isn't important to us right now. Let's focus on these 2 tRNA binding sites and on what happens during the protein synthesis. Once again, you have the small and large ribosomal subunits with the mRNA in between. Here you can see individual nucleotides of the mRNA represented as ovals, and you can see the anti-codon of the tRNAs binding to a triplet of nucleotides, or codon, in the mRNA. Also note the arrowheads in pink on the mRNA, which are meant to show that the mRNA is threading through the ribosome left to right in this illustration. Now you can see all the individual parts. Let's put the system in motion. Each cycle of the ribosome adds a new amino acid to the growing, nascent polypeptide. It starts with a tRNA bound to the P site with the growing polypeptide attached to it. So P is for polypeptide. A new amino acid bound tRNA enters the A site and binds to the next codon of the mRNA. So A is for amino acid. The codon determines which amino acyl tRNA will bind. So the amino acid sequence is encoded by the mRNA sequence, and the code is read by the tRNA. Now you have both the A and P sites occupied. The new amino acid is incorporated into the growing polypeptide chain by transferring the chain to the amino acid bound to the tRNA on the A site. That is, a peptide bond is formed between the amino acid at the A site and the polypeptide at the P site. A ribozyme that is part of the large subunit catalyzes formation of the peptide bond. Now you have the P site occupied by a tRNA with no polypeptide attached to it, and the A site occupied by a tRNA with the growing polypeptide chain attached. Next, the whole ribosome shifts or translocates 3 nucleotides down the mRNA. Before I said that the mRNA threads through the ribosome, but what actually happens is that the ribosome moves along the mRNA. The polypeptide tRNA that was in the A site is now in the P site. The tRNA that was in the P site exits the ribosome, and the A site is empty, and the cycle can happen again. Cycles will continue until the ribosome sees a stop codon, and then the new polypeptide will be released, and the small and large subunits of the ribosome will come apart. Now that we know how DNA encodes protein, we can really look at the molecular basis of genetic diseases. We'll start by talking about mutations. Simply put, a mutation is a change from the normal sequence of DNA. Because they're in DNA, they're propagated from generation to generation, and genetic diseases, as we've already seen, run in families. Mutations take 2 forms: substitutions and insertions-deletions. And when these happen in DNA sequence that codes for protein, the mutation could have an effect on the sequence of the encoded protein. Remember, though, that not all DNA encodes protein. Introns, which we talked about before, do not encode protein. In fact, most of the genome does not encode for protein. Let's start with insertions and deletions in protein coding sequence of DNA. Remember that the genetic code is a triplet of nucleotides. So if you add or lose 1 or 2 nucleotides, you've disrupted that series of triplets, and you've changed all the amino acids that come after the insertion or deletion. In other words, you've altered the reading frame of the encoded mRNA, and these mutations are called frame shift mutations. A frame shift dramatically alters the amino acid sequence of a protein and is likely to lead to a non-functional protein. If 3 nucleotides, or a multiple of 3, are inserted or deleted, then you will gain or lose an amino acid, but the rest of the protein will still be intact. This may or may not affect the function of the protein. It's impossible to predict. A substitution is what the name implies. 1 nucleotide in DNA is changed to another. When this happens in coding sequence of DNA, there are 3 possible effects it could have on the protein. The mutation could be silent, missense, or nonsense. First of all, it may not change the encoded amino acid. The genetic code is redundant. Some amino acids are encoded by more than 1 codon, so it's possible to change the DNA sequence without changing the sequence of the encoded protein. Such mutations are called silent mutations because they have no effect on protein structure or function. A missense mutation changes a codon such that it now encodes a different amino acid. In this case, a nucleotide substitution leads to an amino acid substitution. Whether or not this change leads to a change in protein function is difficult to predict. Finally, a nonsense mutation changes a codon that normally codes for an amino acid to a stop codon. This leads to the production of a shortened or truncated protein that is likely to be non-functional, depending on how much it's truncated. To make sense of all this, let's look at a real-world example, sickle cell anemia. As we've already discussed, sickle cell anemia is a recessive disease that's prevalent among African Americans. Here you can see the mutation in the hemoglobin gene that causes sickle cell anemia. The mutation in DNA leads to a change in the sequence of one particular codon of the mRNA from GAA to GUA, and this leads in turn to an amino acid substitution in the hemoglobin protein from glutamate to valine. It all starts out simply enough, just a single nucleotide substitution leading to an amino acid substitution in hemoglobin. You might not think that this single amino acid change would affect hemoglobin function, but if you look at the structures of glutamate and valine, you see that they have very different chemical properties. Remember that hemoglobin is a protein in red blood cells that binds oxygen and transports it to all parts of the body. The sickle cell mutation changes the structure of hemoglobin such that the protein becomes sticky, so much so that large protein aggregates form inside of red blood cells. This can actually alter the shape of red blood cells to the sickle shape characteristic of the disease. If you look at the picture of red blood cells on the left, you'll see that along with the normal red blood cells, you see some that are sickle shaped. This sickle shape compromises the ability of red blood cells to transport oxygen, and you can see that in the picture at right. The biconcave shape of normal red blood cells makes them very flexible so that they can squeeze through very tight capillary spaces and deliver oxygen to all organs of the body. Sickle cells are misshapen and don't have that flexibility, and don't easily travel through all of the body's capillaries. This explains many of the symptoms of sickle cell anemia. The first symptom is anemia, which means low blood oxygen. The sickle cells are fragile and break down quickly compared to normal red blood cells, leaving fewer cells in the blood to carry oxygen. Another symptom is joint pain. The sickle cells get stuck in small vessels, especially at the joints, causing swelling and pain. This also leads to swelling in the hands and feet. With sickle cell anemia, we've brought things full circle. We learned in our last unit that some diseases are genetic and follow a particular pattern of inheritance. Now we see how a mutation in DNA is not only heritable, but can also lead to a mutant protein which can lead to disease.